The Blacknose dace is a small minnow in the family Cyprinidae. They are commonly found in streams throughout Central and Eastern North America. The eastern Blacknose dace is the subspecies of Blacknose dace most commonly found in the Shenandoah Valley. Turbidity is the measure of particulate matter suspended in water. When water has a high turbidity, this means that the water is not very transparent because of the particles that are suspended in the water. Turbidity tolerance ranges for fish are most often a balance of particulate organic matter that's needed to sustain the fish's prey in the limit of the fish's ability to hunt. Turbid water can occur naturally by means of natural disturbances like increased rainfall, but it can also be caused by human activity. Agricultural practices such as letting livestock wade in creeks leads to erosion, which facilitates the entry of more sediment into rivers and streams. Turbid water can provide security cover for prey as it causes light to scatter. This scattered light hinders the ability of predators to distinguish prey from the background. Thus, turbid water can have differential effects on fish depending on their size, development, and location in the food web. For our research project, we aimed to examine the effect of turbidity on the feeding behavior of Blacknose dace. We chose this species because they are fairly common and easy to capture for testing. We hypothesized that turbidity would affect the feeding behavior of these fish. We went to Dry River with Dr. Krebs to capture Blacknose dace using a shocker. We separated the dace into 2 tanks relatively equal to each other. We separated them by size so that we had a large tank with 15 fish and a small tank with 11 fish. All of the fish were verified by Dr. Boyer to be Blacknose dace. For 2 weeks, we fed the fish daily while we prepared to conduct our testing. Our test consisted of 3 separate trials, with each trial increasing in water turbidity. For the first trial, we did not add any sediment to the water. This was our low turbidity trial. We took samples of the water from each tank to get the exact turbidity value. During the second trial, we added soil to both tanks, and we used a glass cup to try and standardize the amount that we added to each tank. We took a sample of this water as well to measure the turbidity. When we measured the turbidity for these water samples, we experienced a technical measurement error, but it did not affect the quality of the data that we collected. For that reason, we decided to categorize our test groups as low, medium, and high turbidity. For the third trial, we added even more soil to each tank to make it the most turbid water. We reached the desired turbidity and tried our best to feed at the front of the tank so that we could see the bloodworms enter and see the fish that came over to feed. We took a sample of that trial water as well and measured the turbidity. We analyzed our video footage to quantify the number of feeding attempts per tank for each trial. All 3 group members watched each video together and counted how many feeding attempts they saw. In the large tank, when turbidity increased to a medium level, the feeding attempts increased, and then they decreased at the highest turbidity. In the small tank, the number of feeding attempts consistently decreased as the turbidity increased. We had not initially planned to analyze the large dace versus the small dace, but they showed an interesting trend of the smaller fish having fewer feeding attempts than the larger ones. This may be due to the fact that there were less dace in the small tank than in the large tank. It could also be due to the fact that the small dace had smaller mouths than the larger dace, so it took them longer to process the bloodworms, which were the same size for both tanks. This finding may be supported by Utna Palm's 2002 article, which states that turbid water tends to be more beneficial for younger, smaller fish in terms of security cover and less beneficial for larger fish that prey on others. Additionally, from this article, it was shown that turbidity can hinder long-distance food detection, but it has little effect on their ability to detect nearby objects. We saw this trend with our large dace when they showed more feeding attempts in the medium turbidity trial. With another article, Madej 2002, they found that increase in turbidity affects the efficiency of fish feeding, but that a limited amount of fish feeding continues at moderate turbidity levels. As human encroachment on freshwater habitat progresses, it will become more valuable to understand how increased turbidity levels affect the fish that live in the waters.